Hello, everyone, and welcome to today's webinar titled Take a Leap Forward in Digital PCR Technology with the Roche Digital LightCycler System. This webinar is a part of the 14th annual event in the Clinical Diagnostics and Research Virtual Event Series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Roche. For more information about Roche, please go to www.roche.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You will also notice that the speaker has provided a survey located on the right of your screen. Please take a few moments to answer the survey questions. It will be available during both the live session and on-demand period. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the event. All right, now let's get started. I'd like to now present today's speaker, Simran Saini, Principal Scientist 2, Sequencing Workflows, Applications, and Technology at Roche. For a complete biography on our speaker, please find it on the left of your screen. Simran, you may now begin your presentation.
First and foremost, again, thank you for attending today's session. I'll be introducing the Roche Digital LightCycler System. Again, a powerful new addition to the Roche PCR ecosystem that will really enable users to unleash the true power of digital PCR. So, as many of you are aware, over the last decade, digital PCR has emerged as the next generation of PCR technology because it permits absolute quantitation, which gives it significant potential to impact a variety of clinical research fields, right? For instance, oncology, as well as infectious diseases, amongst others. So while there are existing dPCR systems on market, we at Roche really wanted to design a dPCR system that would really enable users to push the boundaries of their research forward by filling in the gaps and making improvements upon existing offerings. So with that in mind, we asked primary users of digital PCR technologies what they would look for in a digital PCR system if they could actually design one. And the feedback that we received essentially was that they were looking for something that had IVD clearance, something that had reduced grain, which is essentially enables very clear-cut thresholding. Remember, digital PCR is a binary. It's essentially the presence or absence of something. So the capability of being able to classify positive and negatives is paramount for digital PCR. Additionally, there was a need for increased system flexibility, but really wanting a system that's flexible, that can be tailored to specific applications. Obviously, lower dead volume, which again speaks for itself, but just to highlight here, you have the capability of measuring everything that you're actually putting into the system, and greater multiplexing capabilities. Again, there's 6 different channels that are located on this system that allows for a great multiplexing capability on this system. So again, we took this feedback that we solicited and wanted to input and design this into a digital PCR system that would actually exist within our PCR ecosystem. And again, for those of you that are not familiar with Roche Diagnostics, Roche was an early pioneer in PCR and has really remained at the forefront, right? We have helped to advance and improve PCR technologies and assays for decades, so this is not something new for us. These technologies include the digital LightCycler 480, which is actually a qPCR-based instrument, paired with the MagnaPure systems, which are essentially used for automated nucleic extraction, and can be combined with the LightCycler technology essentially within our CoBOS offerings. And so, as you can see, that pioneering legacy has actually kind of moved forward and continues onward with the addition of the digital LightCycler system, where we've incorporated a unique combination of technical advancements, the digital PCR technologies, compared to what's on market. And that unique combination includes 3 different nanowell plate types, something that I will be describing a little bit more in closer detail, but just to kind of visually show it right here, this is a nanowell plate, which we have 3 different configurations of. Additionally, there are 6 advanced optical channels, but there's actually also an additional channel which does not allow for content, but is utilized for detection of partitions that are filled with a fill control that is essentially located in the 5x master mixes, which you can imagine is very important, right? You only want to assess partitions in digital PCR that actually have been filled appropriately. In addition to that, we have the 5x concentrated DNA and RNA master mixes, which have been essentially optimized for multiplexing and also enable very much having a lot of sample input because they're highly concentrated. So these technical advancements have been integrated into the overall digital LightCycler system, which is comprised of the following components. As I mentioned, there's the consumable, which is this nanowell plate, and just to kind of walk you through this, but this is essentially a nanowell plate. In particular, you can see there is a barcode that's associated on the plate itself. There are 8 inlet ports here, so you can imagine a sample per lane. Each one is designated as a lane here, and then the output lane right here, essentially. So this is kind of the anatomy of the consumable. But as I have kind of briefly touched upon, there are 3 different configurations of this, and that's outlined here. We have the high sensitivity, which allows for a 45 microliter input load with 20,000 partitions. Remember, these are physical partitions that are imprinted on the lane itself. And you can imagine this is really well tailored to cell-free DNA applications, oncology applications, as well as for rare mutant detection, where you really want to sample as much as you can, right? You want as much sample to assess. And with the 45 microliter input, that does allow you to do so. Then we have another configuration of the nanowell plate, which is the universal. This has a 30 microliter load with 28,000 partitions. Again, very much tailored to applications that are specific, potentially for things like gene expression and or transplant rejection. And then the last kind of configuration is the high resolution. This is my favorite consumable because it has 100,000 partitions per lane with a 15 microliter load. And you can imagine that it's well attuned to things that require high precision, like copy number variation, NIPT, and also for human genetic diseases. So these configurations, again, all supported by the digital LightCycler system in this nanowell plate configuration. In addition to the consumable, there's also a standalone partitioning engine right here, which enables essentially sample distribution across the entirety of this nanowell plate. Then we have the analyzer, which essentially has 6 optical channels, again for content, enabling multiplexing. And finally, the generic DNA and RNA master mix. As already mentioned, it's 5x, highly concentrated, affords a lot of sample to be input into the reaction itself for measurement purposes. So now that you're a little bit familiar with the system components, I'd like to actually walk you through a workflow. And I'm going to kind of be focusing on the diagnostic workflow. So in this case, you can imagine, for instance, that you have samples that needed to be run. And what you need to do originally in the diagnostic workflow is to kind of use that input of the samples that you'd like to run in the batch, for instance, through the web application. Now, this can be done manually, or it can be associated from orders that come from an established LIS system. And what happens is that these particular samples are inputted and associated with the consumable. As I mentioned, the consumable itself has a barcode that's attached, right, to each plate. So you can imagine that association occurs via the web application, and therefore, at the outset, you already have sample tracking that's occurring. Next, the extracted samples are combined with the digital PCR mix, primers and probes, essentially, and then pipetted into the actual nanowell plate based on the assignment that's been given by the web application. Once the nanowell plate has been loaded, it is then partitioned within the standalone partitioning engine, which also scans the nanowell plate and monitors the partitioning process. There's essentially tracking throughout this workflow in terms of also looking at it from the perspective of the system level. Once the nanowell plate has been essentially loaded and partitioned, then it is placed onto the analyzer itself. And what you can see here is that there's up to 12 plates that can be placed on the analyzer in a given run. And again, 8 wells per plate, meaning that you have a flexibility of a batch size from anywhere from 8 to 96 samples in a single run. Additionally, there's the added flexibility that you can run multiple plate configurations in a single run. So you can run 2 universals alongside 2 high-res, so long as it's under the 12 plate requirement for the system itself. Now, once this is accomplished, the analyzer also will scan the barcodes for the plates that have been inloaded and will essentially permit that sample tracking to occur continuously throughout the process. And after the run is complete, the results are validated essentially and then sent back to the LIS. Okay? So when that has been accomplished, then there is an opportunity essentially to go through and use either the development software that would be more for research-based focuses, or having analysis packages that are already predetermined to allow for results to come out from your run itself. So one of the really interesting aspects of that workflow is the partitioning. And so I wanted to kind of spend a little bit of time focusing on that aspect. So just to kind of a little bit more highlights of the partitioning engine itself, here are some of the key features. First, it is operational by touchscreen. It also has smart liquid handling of the partitioning fluid, which essentially means that it tracks how much of the partitioning fluid is present in the system, whether or not you need to change. It'll prompt you based on the touchscreen and allow for you to kind of do that in a very seamless manner. Now, one other thing to kind of really highlight is that it's standalone. Although it is connected to the analyzer in the sense that there is relay of information, for instance, whether the plates have been partitioned and whether the analyzer can recognize that partitioning to make sure that it doesn't run plates that have not been partitioned. That all essentially is integrated into the connectivity between the partitioning engine and the analyzer. However, there is the opportunity to have the partitioning engine as a standalone in a different room, for instance, which then could be connected through the local laboratory network. You can imagine that this is really a nice feature, given the fact that you may want to have the partitioning engine in a pre-PCR lab, for instance, where you have no amplicon generation whatsoever, and the analyzer residing in a post PCR lab where there's amplicon generation, right? So there is that opportunity and flexibility to have them separated, or they can also remain connected in a single room configuration. So very much lab specific. Now, how does the partitioning itself actually occur? So what's being shown here in kind of cartoon version is a side view, and right below it is kind of a top-down view. And let me just kind of walk you through. As I mentioned, in the nanowell consumable itself, there are 8 inlet ports. And so initially what happens at the outset is sample is pipetted into each of these wells in a manual way, right? So it's a passive flow that will occur. And that's what's being demonstrated here in these first 2 kind of diagrams. Here you have a pipette going into the inlet port. Again, depending upon the consumable type that you're using, there are different fill volumes. But effectively, it'll start to migrate due to kind of capillary action about 1/3 the course of the length. Now, afterwards, this plate is then placed one at a time into the partitioning engine unit that essentially will also input oil, okay, or partitioning fluid, let's just say. And that partitioning fluid will get disseminated by a pressurized base system in the PE itself. And then as the kind of pressurization mechanism it gets employed, it will move both the sample as well as the oil across the length of the lane, effectively sealing it all together entirely. So one thing that I want to kind of highlight here is that sealing essentially ensures that there's no amplicon kind of contamination that occurs, right? It's a closed system. You have complete sealing of the entirety of the lane, right? Which is really, really important when you're trying to contain amplicons that are being generated during the digital PCR or PCR process itself. So now that you're more familiar with the digital LightCycler system, kind of walked you through the workflow as well, I just want to kind of address, as we mentioned earlier, how the Roche Digital LightCycler System actually represents a step forward, right, in dPCR performance by focusing on 4 key areas where we've kind of seen advancements with this particular system. And that includes sensitivity, precision, flexibility, and integration. Hopefully some things that I've already kind of touched upon, but kind of just really want to call out and also showcase some data around. So let's start with sensitivity. So as I mentioned, sensitivity can be very critical, right, for certain applications. So in this instance, we're going to highlight rare mutant detection on the digital LightCycler system because it has the enhanced capability of really detecting minimal copies of DNA and RNA, and for the following reasons. So we have the high sensitivity nanowell plate, which is specifically tailored to have 20,000 partitions and a high fill volume, right, 45 microliters, which essentially ensures that you are assessing as much sample as possible, which is really important when you're looking for a rare mutant. In addition to that, we have the 5x concentrated master mix for both RNA and DNA. Again, affording a lot of room for sample input as opposed to having a lot of kind of the reaction volume made up from the the actual master mix itself, right? So kind of just making sure that there's an opportunity to sample as much as you can during the course of this particular application. In addition to that, there's very little rain and clear cluster separation, which means, again, easier thresholding. As I've already alluded to, digital PCR is a binary thing. Either you have the presence or absence of signal, and the capability of thresholding will enhance your resolution in terms of classifying both positives and negatives, which is very critical. So I just wanted to kind of walk you through one example, and this is going to be an oncology assay detecting low-frequency single nucleotide mutation in BRAF. And so kind of bear with me here. But essentially what's being shown here is an input sample of cell line genomic DNA at 150 nanograms per reaction. Here on the x-axis is essentially the titration of a mutant from 1% to about 0.031% with constant wild-type DNA background at 1,500 copies per microliter. So what I want to kind of highlight here is that in blue is the wild type. You can see, very much equivalent loading across each of these lanes here, as you can see with the number of copies that are depicted. And again, on the x-axis is that mutant kind of titration, all highlighted in red. So the closed circles are essentially the replicates, so the actual measurements, and then the lines are essentially the confidence intervals, both the high and the low, respective of the replicates that were run. And just kind of focusing on the wild type in particular, what I'd like to kind of showcase here is that we had observed noise in the system with the 95% confidence interval upper limit set at 0.11 copies per microliter, which is kind of denoted by this dashed line here. And that was used to establish the cutoff, essentially. So what we're trying to say is that anything that exceeds the cutoff that was established by the wild type, which essentially did not have any mutant, would effectively be called above noise, right? So that's what we want to kind of showcase here. And so hopefully what you can appreciate here, going from the low end to the high end from that titration, is that at about right here, 0.063%, you had very, very nice and clear and significant separation above the cutoff, which is essentially set by the wild type. So really, really nice, tight replicates, confidence intervals very much above that. And also just wanted to kind of showcase a few of those plots and to show you what the data actually looks like on the system. So just coming off of that from the numerical quantitative perspective, now just kind of looking at 3 of those data points from the plot perspective. These are 2D scatter plots representing the 0.25% in that previous slide, the 0.63, as well as the wild type for comparison purposes. So in each plot, the brown cluster is composed of partitions without the template, so essentially your complete negatives. The blue cluster is the wild type, the orange cluster is the mutant, and the green cluster essentially has both wild type and mutant, so your double occupant, partitions effectively. And what I'd like to, and hope you can appreciate, between these plots is that there is significant difference between separations between each of these clusters, right? There's a capability of thresholding very nicely across these kind of conditions, which allows for a really nice, accurate quantification overall. And more importantly, I'd like to kind of indicate that the system is able to detect, that the BRAF V600E mutation at less than 0.1% allele fraction, right? Which is again depicted by this center plot right here when compared to the wild type. So again, very nice capability based on the attributes that are built into the system to have really low detection of rare mutants. I also want to kind of indicate, in addition to the rare mutant detection capability, the digital LightCycler system is also very well attuned to detecting indels, and that's insertions, deletions, down to less than 0.2% allele fraction. So kind of looking and walking you through that. Here again, what we're showcasing is cell line genomic DNA at 66 nanograms per reaction. The titration of the mutant is from 0.063% to 1%, again, obviously including wild type, which has no mutant titrated within. The negative control, this is, I should have mentioned the assay, my apologies. This is an EGFR exon 19 deletion mutant that we're looking at here. And very similarly for the rare mutant detection, we just wanted to look at the wild type to set the cutoff based on the noise that was observed. In this case, it's the confidence interval that sets that cutoff, and we're looking at about 0.23 copies per microliter. And so what I hope you can appreciate again is, based on the cutoff, that we have a sample here, two samples, two replicates at 0.125% that can be significantly called over that established cutoff from the wild type. Again, very nice resolution, really great separation. And again, just wanted to showcase what the actual data looks like coming off of the system. Again, 2D plots are being represented here for the 0.25% EGFR exon 19 deletion, the 0.125% titration, and obviously wild type for comparison purposes. And just again, very similar to the last plots, the brown cluster here is comprised of no template, so essentially your complete negatives. The green clusters here are partitions with wild type template as well as wild type plus variance templates. And the blue cluster here, which is kind of highlighted and circled in orange, is your mutant, essentially. And again, you can see really nice titration going down between 0.25 and 0.25 just by the number of mutant copies that you see kind of coming up here with the blue representation. But again, very much want to highlight that above the wild type in terms of the background, you see really, really nice kind of formation of clusters that are easily thresholded between. So now I'm going to move on to precision. This is, again, a very key advancement, at least in terms of the digital LightCycler system, to allow for a high level of precision, which is really important for looking at measurements of small quantitative differences, right? Really, that's a hallmark, for instance, of copy number variation assays. And so, again, what are the features in the digital LightCycler system that allow for this kind of high precision measurement? Well, one thing is the consumable, which is, again, one of our particular configurations that are offered, and that is the high resolution plate, which has 100,000 partitions per lane. In addition to that, we have 6-channel detection capability. You can imagine for copy number variation, there obviously needs to be a reference that's utilized. And with the number of channels that are afforded to you on the system, there's an opportunity to use more than one reference. And so why is that important? Why is having more partitions important? And why is having the capability of running multiple references important? So let me kind of walk you through that. So this is a very busy slide, and it can be a little bit hard to see, but let me just kind of focus your attention first on the axes. So on the X, what we have first shown in this kind of pairing of numbers that is separated by a comma, the first number essentially represents lambda, or the copies per partition, effectively. And that's kind of followed by a sample input in a nanogram per reaction kind of metric. So just to give you an idea of, how much of DNA we're working with, or RNA, for instance, that's just kind of what's depicted here on the x-axis. And it's going from the lowest amount here, so at a copies per partition about 0.01, which translates to about 0.91 nanograms per reaction, all the way onto the far end, where we're looking at the more ideal number of copies per partition, which is about 1.57, 1.6, and then that's translating at least to about 143 nanograms total in the reaction. Now on the y-axis, what we're looking at is diploid copy number, right? And so what I really want to point out is that we're looking and going to be using as an example a copy number variation between 10 and 11. So 10 is depicted here and 11 here. 10 is represented by closed circles, and 11 copies is kind of represented by closed triangles. And what's emanating from both the circles and the triangles are the confidence intervals, essentially. Okay, both the high confidence interval and then the low confidence interval. And the reason there are two colors is because of the number of references that are used. In the case of the blue line, we're looking at one reference. In the case of the red line, we're looking at two references. So now what I'd like you to appreciate is that when we're looking at the number of partitions, in this case 28,000 partitions on the universal configuration of our plate type, you can see that as you start to look at separation, where there's no overlap between the confidence intervals, which essentially means that you have confidence in detecting that copy number difference of 10 and 11, it's very limited, very, very small dynamic range that's only kind of situated here and here. Okay, so very, very small interval where you have non-overlapping confidence intervals, which means that there's a capability of resolving 10 copies from 11 copies. Now, if we move to a different scenario where we have 100,000 partitions. And again, this is the same kind of graphical representation in terms of the axes, the X and Y axes, so I won't go over it again. But what I hope you can appreciate is that there's a wider dynamic range of gray space right here, where there's, again, non-overlapping confidence intervals, which means that for a larger range of inputs, there is a capability, essentially, of resolving 10 copies versus 11 copies. So really high precision afforded by the fact that you have more partitions that you are capable of assessing. In addition to that, it's not something that maybe could have been picked up, and so I'll kind of walk back into this slide just for an easier visualization. Depending upon the number of references, you can see that confidence interval also starts to shrink. And so maybe I'll just kind of point out this region in here in particular, where you can see that when you have one reference, the confidence intervals are a little bit longer than when you have two references, right? And so that also translates, in this case, very similarly to the high-res consumable, and also the fact that this particular instrument has multiple channels that it can utilize multiple references. So you can imagine those confidence intervals can even shrink further the more references that you end up using, again, for very precise measurement applications such as copy number variation. And here's an example. Just wanted to give you an actual data example of a copy number variation assay that was tested on the digital LightCycler system that was using two different cell lines that differ in their copy numbers, again, 11 and 10. As you can see here, what we did is we used pre-digested genomic DNA at 175 nanograms per reaction. Here are the two cell lines. Again, one that is having a copy number of 11, one that's having a copy number of 10. So what's being shown here on this plot here is the copy number. What we've kind of put as a benchmark for ease of visualization is the copy number 10 here grayed out and copy number 11 here on top. Again, the two different cell lines, so the copy number 11 represented by blue, the copy number 10 represented by red, and kind of also bracketed by the confidence intervals, both the low and the high. And again, quantitated for you here, both the copy number as well as the minimum and maximum confidence intervals, just for ease of visualization. And what I hope you can appreciate is that there is a capability from the replicates and with the cell line that was tested that the resolution between 10 and 11 is very, very strong, right? There is a capability on the system using the high resolution plate to be able to differentiate very small differences in copy number variation. So now I'm going to move on to flexibility, which is kind of our next pillar in terms of advancements that have been inbuilt into this system. And so some of these have already been touched upon, but again, just wanted to highlight that here. The digital LightCycler system is a very flexible clinical research tool with an ability to choose different batch size increments, as I mentioned, anywhere from 8 to 96 in a single run. There's an ability to combine lanes for a single sample to increase resolution. So for instance, if your application required that you merge, 100,000 partition lanes, you can do that easily in the development software. So there's a capability of kind of harnessing the development software that does the analysis as well in terms of aggregating and analyzing data. There's an ability to really kind of track samples, as I mentioned with the workflow slide, that there is a barcode that is associated with each of these consumables that gets tracked throughout the entirety of the workflow. Plus, there's the added capability of interacting with an associated, kind of LIS system and or using the web application as well. So very much complete sample tracking at the outset. There's also a capability of doing high multiplexing with the 6 optical channels, as already demonstrated with both DNA and RNA reagents. And you can also address various application needs, some of which I've already walked you through, based on the configuration of the consumable that you'd like to use. Whether it's the high sensitivity for rare mutant detection, or it's the high resolution for things like copy number variation. So another thing that kind of falls within the flexibility is actually sample compatibility, which I kind of illustrated on the workflow slide. I kind of had those different sample types listed, but I really wanted to call that out here. Due to the fact that we're using actual physical partitions on these consumables, as opposed to kind of emulsion-based droplet generation, there is actually a capability of being a little bit more robust to things that carry over, for instance, from nucleic acid reaction that might have some impact on the chemistry that's involved in kind of creating droplets, for instance. You can also imagine, and this is kind of a side note, not necessarily tailored to samples, that in the ability to kind of utilize the system, you take that variability out, right? These are fixed physical partitions that are present on this consumable, and so you don't have to worry about the added variability in terms of users being able to generate the optimal amount of droplets, for instance, in a given run. So it's a really nice kind of system that's been built up. But, kind of bring it back to the sample types. We are compatible with a variety of samples, FFPET, cell-free DNA, whole blood, eluate, stool, as well as swab. And again, there is a capability even from the samples themselves, which have intrinsic properties that do kind of have limitations potentially in terms of being robust in, for instance, qPCR because of carryover or intrinsic properties that necessarily would not be the case on the digital PCR platform, given the fact that we're using physical partitions as well. Additionally, under that kind of flexibility bucket, I do also want to kind of call out the software, which we haven't spent so much time on, but it really takes out the frustration of sample tracking, as I mentioned, as well as data collation and analysis of your results overall. And that's because it has a lot of different features, right? There's remote accessibility to the software. There's automatic clustering that's employed in the development software. There's a capability using the software itself, to kind of develop your own assay analysis packages that are tailored specific to your needs. You can build your own lab-developed tests and actually have that as a designated analysis package that you use, going forward for any type of LDTs or things of that nature. And already built into the development software, there is different analysis types and packages that are already afforded into the system. For instance, absolute quantification, looking at indel analysis, as well as copy number variation. Full sample tracking as already highlighted, and also the ability to interface with the LIS system that's already established in the lab. And now to the final pillar, which is integration. So as I mentioned, the digital LightCycler system has all of these really great features, and it actually also integrates them very seamlessly. And I know that these have already been highlighted, but I just want to kind of summarize it here because it's really the system overall that really demonstrates and outputs the data that's of very high quality. And that's because we have, the chemistry of the generic reagents, which includes of the 5x concentration master mix. Again, very much highlighting the need to have as much sample input as possible. The DNA and RNA have both been optimized for multiplexing capability. There's a fill die control, which validates partitioning. As I mentioned, you don't want to have empty partitions that are also calculated in your actual quantitation methods. This is done on a Poisson distribution, essentially. And so those are actually taken out of consideration, really allowing for high precision measurements overall. There are 6 optical channels. There are 3 different nanowell plate types. Again, a closed system minimizing the chance of amplicon contamination, given the fact that the partitioning process itself effectively seals each of these individual reaction partitions. There's also the ability to connect to an established LIS system, very much improved for digital kind of workflow and sample tracking overall. And the algorithms that are built into the development software are really kind of been very much kind of attuned to rejecting non-PCR artifacts, right? So we're not measuring things that are inappropriate. We're looking at things that are only relevant to the sample itself and overall for your actual output for your data. Absolute quantification, without the need of standard curves, which is a hallmark for digital PCR, and very much makes it a step above, qPCR-based technologies. So really what we're trying to do is we're enabling laboratories to really explore their research, knowing that they will generate, high quality results, at the highest level of confidence, right? We're kind of building into all of these kind of, extra amenities essentially to allow for very, very trustworthy results to come out of this system. But at the end of the day, what it really matters is your data, right, and your results. And so just wanted to kind of highlight that integration on the digital LightCycler system compared to 2 other platforms that are on market as well. So what's being shown here essentially is the 3 different systems that are running what we call a valid prime assay. This is a commercially available assay. On the very far left here is the Roche Digital LightCycler System. Here in the center is the NICA dPCR system, and then on the far right is the QX200 digital PCR system from Biography. And what I can hope you can appreciate from looking at these 3 different scatter plots on these 3 different systems using the same valid prime assay is that the Roche Digital LightCycler system has really, really nice cluster separation between the positives and the negatives. It has a tighter positive cluster and a tighter negative cluster, and also very little range compared to the other 2 systems. And this is very important in terms of us allowing for really clear thresholding, again, really wanting to harness digital PCR's technology, which is essentially just looking at classification of positives and negatives. And all of these attributes, that is demonstrated clearly using this valid prime assay across these 3 systems, essentially ensures that there is a nice capability of separating positives and negatives, really instilling that your data that you're actually getting out of the system is of high quality. So, there's also wanting to kind of highlight that this system is essentially allowing for and suitable for a variety of applications, right? And I'll kind of walk you through a couple of those on the next slide. So again, as already mentioned, oncology is really a big field. Obviously, looking at variant detection, microsatellite instability, pathogen for infectious diseases, transplant, whether it's graft rejection in transplant cases, environmental surveillance, large clinical trials, and also NIPT as another attribute. And the digital LightCycler really fits, based on all of the attributes that I've kind of walked you through into all of these different applications. So we really, really urge you, to kind of make that leap with digital LightCycler system from Roche and unleash the true power of digital PCR.
Thank you, Simran Saini, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today as well. I'd also like to thank our sponsor, Roche, for sponsoring today's webinar. Questions submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.